cool. Howdy, folks. So today we are going to be talking about brains, and, well, usually this lecture corresponds to about Halloween, a little earlier, at least October, so we'll see. But anyway, so this is the Biology, Anatomy, and Physiology unit. This is the one that y'all pre-meds, you seem to like this one. So yeah, that's where we're going today. Yeah, cool. Let's just get started. This is going to be a bit of a long unit, kind of similar to how like the personality development unit and the research methods unit, those tend to be pretty long. Sorry. That's going to be the deal this time too. But that's because there's a ton of stuff. Even though I have boiled it down to like 2 objectives, these are pretty big, steep objectives. The first one is discuss what the structure or anatomy of the brain can tell us about personality. And 2, we're going to discuss how personality is related to biochemistry. So those are the big 2, and within each of those, there's going to be lots of little sub-aspects. So this video is getting broken up a lot, which I think is good. You'll let me know. I'm very confident I will be told if it's awful. So yeah, the 2 big aspects of the brain that can be examined with tech are the anatomy. I realize, I feel like I just said that, but I'd rather be kind of concrete. So the anatomy, which is like which is all about the functions of the parts of the brain, and second is the biochemistry. So those are the effects of neurotransmitters and the hormones on the brain process. So both of these are related to personality and behavior, but we're going to kind of break them down. So the anatomy ones are like the physical brain pieces, and the biochemistry are like the ways the brain pieces talk to each other. And you may notice I'm going to use a lot of informal language. I am not a neuroscientist by training. I've played with neuroscience data, but I apologize in advance that this is one of my even though I study like the genetics of personality and stuff like that, the biochemistry aspect is pretty distant. You can ask me all about Mendelian randomization, and I got you covered. So, basic parts of the nerve. I've got some figures floating around coming up. So you got the dendrites, and these are the projections on nerve cells that receive stimulation, and the axons, which pass that stimulation from the nerve cell onwards. I'm gonna attempt to, oh god, this is gonna be awful. I hope you enjoy. Okay, so we got the, like, axons, whoosh. Ooh, this is terrible. What? Okay, I'm gonna keep this in and include an overlay right here. Wow. I don't even know what, in my mind, that is a very beautiful nerve. Clearly, I leave a lot to be desired in this drawing. So I'm gonna cut in and include a nice overlay talking about the dendrites and the axons and those 2 parts of the nerves. Now, once we all have a clear image that doesn't look like a broken guitar where all the strings burst at once. Wow. Sorry, it looks way better when I hand draw it, relatively speaking. And, oh, Tooks, come here. I'm sorry, I gotta pause here, because Tuki's still in his harness, and he's He's not liking it. All right, I'll be right back. Okay, I'm back. Sorry. So graceful. So I had tried out Tuki's bigger harness because he's close to a big boy, and it works. It's effective, but I got distracted, and so I left him in it, and he was so tolerant that he just kind of, he's not a huge fan, he tolerates it, so I can go outside. Okay, back to nerves. So we got two types of nerves. We've got afferent nerves, and these are where the message travels along the nerves from the body to the central nervous system. So it goes kind of outside in. Then we have efferent nerves, which are messages that travel across these nerves, go from the central nervous system to the body, so they go inside out. And lastly, we've got interneurons, which kind of organize and regulate the transmissions. Oh, I thought there was an extra word there. There's not. Transmissions between nerve cells. The biggest bundle of these are in the brain. Cool. Yeah. So, next. I'm going to include this brain slide a lot because while I'm not going to assess you on being able to map the different parts of the brain to the different labels, I find it helpful to imagine them. And this is figure 8.1 in the book, and I'll go through it here, but it'll keep popping up. Hello. Hi. Nope. But anyway, so and you've hopefully covered a lot of this in intro, and you'll cover it in way better detail in any neuroscience or biochemistry of the brain class. So the hypothalamus is connected to just about everything in the brain. It's this little pinky blob here. So it secretes several hormones that we'll talk about. That's kind of the brain chemistry side. Next, we got the amygdala, which in this diagram is this green little pea-shaped or pea green color thing. It's got an important role in emotion. We'll talk about this one later. Again, this is more of like a overview of here are the parts we're going to talk about. Got the hippocampus, which is this blobber right here. Its focus seems to be important to processing memories. We got the cortex, that's the whole outer layer of the brain. The neocortex is even the outermost layer of the cortex and is the most distinctive part. It's the wrinkly part. We've got the frontal cortex, it's large. You know this. So it's the frontal lobe. It's right in the front. It's large, it's really crucial to a lot of uniquely human aspects of cognition, such as planning ahead, anticipating consequences, and emotional experiences. And I've got some other pieces pointed out here. The corpus callosum allows the two halves of your brain to talk to each other. And while there is some truth to that left brain, right brain thing, it's not as personality based. There still are specializations on brain halves, but it's not like you're fully dominated by one half if you're by the right half if you're more logical, or in the left half if you're more creative. But it's a cutesy way to think about it. Alright, so here are a couple of the classic, oldest school research methods for studying the brain. Are brain damage. Obviously, we don't induce these in people intentionally. Usually we look at the various problems that are caused by damage to different parts of the brain. We're going to talk tons about Phineas Gage. I have very strong opinions. And if we were in class, I would have a prop to give you a sense of how big the iron spike is. It's like armpit level on me. It's huge. But, so, Phineas Gage is a classic example. We'll talk a lot about him. So there is some lab research done using brain damage in non-human mammals. This is primarily on animals. This is study. So like a lesion is like where you essentially damage part of the brain and you see what happens. And so the study of animal personality is a growing field, and it kind of falls into this brain lesion work here because there are animal characteristics that I don't think I talk about anywhere else in this lecture. But I'll bring in some cat examples in later units. But yeah, so let's just keep going because I know that you guys want to move on and watch the next lecture. So let's keep my little asides. It's mid-afternoon, and so that's why I'm a little lower energy right now. But yeah, so brain damage, you break stuff, you see what happens. Brain stimulation, way less invasive and not as permanent. The idea is that you stimulate the brain directly to see what happens. So like the electric signals, you send them in, and we see what happens. Now, this is pretty difficult and rare to do in people while conscious, but it is done in people on occasion. Usually when a brain surgery is being done, you check stuff to make sure it's still working. And I've got a cool kind of illustration on the next slide, illustrating one woman's experience of an acute depressive episode based on just brain stimulation in this area deep in the midbrain. It's the substantia nigra. I don't know how it would be pronounced in English, but in Latin it'd be substantia nigra. Of one woman, and it's figure 8.2 in the book. And so that's one piece. Next, we've got some transcranial magnetic stimulation as well as transcranial direct current stimulation. So these are used to create virtual lesions. So by turning on and off parts of the brain to see how various psychological processes are affected. Now, the idea with most of these brain stimulation type research is it's a less permanent version, and you don't have to just capitalize on like weird brain damage to understand your study. You could almost argue it's the classic version. It's like a kind of a neat quasi-experimental design where you kind of capitalize on a natural random event and just kind of crawl deeply into it. Cool. So here's that figure that I was talking about that illustrates how you can induce acute and passing depression. Acute meaning like rapid onset, and transient meaning it's passing through, like it turns off. It's fleeting, it's passing from high frequency deep brain stimulation. So the fact that you can trigger a really strong emotional response from electrical signals kind of indicates that is a key aspect of mood, and it can impact large aspects of how we think, feel, and behave. So next time, I'm trying to keep these pretty short because there's a lot. We're going to talk about research methods for studying the brain. Shocking that the research methodologist wants to talk about the research method. I find this really helpful just to get a sense of like what all the different brain study stuff are, because they do different things, and after a while they start to sound really similar. So I find it really helpful, and actually the act of like making this lecture and really getting a hold on like all the different methods and like looking at big pictures of them and like was really helpful to me to like, because I had some good sense of the different kinds of methods, but it was really helpful and useful to me to just like see them and like actively engage and kind of like consolidate the material. So you get to enjoy the fruits of that labor. I will see you all next time. Until then, Bye-bye.